and I will present to you today mainly the work of Arnon Matza, who wasn't able to come himself, and I'm actually a wet lab biologist, so I hope I can answer your questions. Okay, and the work is an integral programming framework for inferring disease complexes from network data. And the idea behind this is, if you have a disease and you have a known set of genes, you can use it in a network to add genes to your data set by using a propagation algorithm, where you push flow from a priors into the network, and not only the first degree neighbors are then getting a high propagation score, but for example also here P5. So how can these networks be shaped? They could be annotation based, so for each gene you know that there are, for example in Go, biological process, molecular function or cellular component, and this data could be used to build a network with edges. Also Höhendorf et al., for example, he presented a phenotypic-based network where he looked in mice and the effect of knockout of genes on the phenotype, extrapolated it to the human homologue, and built a network out of that. But we want to focus today on protein-protein interaction networks, where the baseline is that genes that lie close in a network also are predicted to cause similar phenotypes and are also maybe associated with a disease. So many studies link diseases to dysfunction of proteins assembling, working in process. So proteins don't only work as a single molecular machinery, they work in a concert with other proteins. These other, only together, they can then sometimes make it function. For example, in Lee syndrome affects the respiratory chain in mitochondria, and the respiratory chain is a very huge complex of many proteins. And these mutations can occur in different proteins of this respiratory chain, which all lead to the disease. So this in mind is the reason why we do this. Methods that are using this propagation are up to date PRINCE and HotNet, for example, and I will come to that also later. So first of all, our workflow. What we needed first, a protein interaction network, and also a set of priors to define our for the diseases. And after we had that, we then did the propagation, as I said before. And when we did the propagation, all the proteins in the network have a propagation score. And then we had to do a thresholding, so make a cutoff, which proteins are included in our subnetwork, which we then put in for the clustering process. For the protein interaction network, we used the HIPPY database, and this is one of the largest protein-protein interaction databases, and in this it contains nearly 150,000 medium or high confident interactions over nearly 15,000 nodes. This confidence score is based on how often a protein is found, interaction is found, and with which method. And then for the priors, we used 3 different databases: OMIM, ORPHA data, and diseases. And there we looked we took only the manual curated genes associated with disease, and not the predicted or experimental database ones, and we used disease ontology as a common dictionary. So there we faced a problem. For example, ALS was in one database only named ALS and had several genes associated with it. In other databases, there were 10 subtypes of ALS, all carrying only 2 genes. So what we did, we did compute that we go upstream of a name until we had at least 10 genes per name. And then afterwards we removed two small terms and handled redundancy and came up with 114 diseases. Then we start the propagation, and this follows actually quite similar the method of Prince. And so the priors push flow to the network, and then high-scoring proteins are likely to be associated with a disease. But, for example, Prince is a method where they arbitrarily choose a cutoff. Say like, okay, after a threshold of 0.03 for propagation score, this is considered associated. We choose a different strategy. We use statistical filtering. And the reason why is the propagation score highly depends on the prior size. So if you have a prior of 100 genes or proteins, you will have a lot higher propagation scores in the network than if you have only a prior size of 10. So with the Prince method, you maybe would overestimate the numbers of interesting proteins for large prior sizes and underestimate it for smaller sizes. So what we did is we took our network and we performed 1,000 random propagations, so for each prior size. So we took prior size 100 and executed 1,000 times 100 with random priors. And then in the end, all nodes have a random propagation score. Then we execute with our prior set of interest and compared if the propagation score of for our prior size is significantly enriched. And only those which are with a significance of 0.01 would be included in our subnetwork. And then, where Prince used a heuristic method, we used an integer linear programming algorithm where we wanted to look at very dense clusters. So therefore, we reweighted the edges of the network. And then we took not the subnetwork, but the whole network, and looked how likely it is that every edge appears in a random network. And why do we do this? If you imagine, if you have a protein with only 3 interactions and it came up in a cluster, it's more likely to be relevant than a protein that has maybe hundreds of interactions and came up in a network. So here, edges get a higher weight if the proteins have less interactions. Additionally, we also assign negative weights for non-interactions. Yeah. Additionally, we forced our clusters to be connected, that each node is at least connected to half of the other nodes in the cluster, and we choose to cut the range of cluster size to 4 to 20 nodes. In comparison to Prince, as I told you, Prince uses fixed thresholds for the propagation score and a heuristic clustering algorithm. Hotnet, on the other hand, they call it not propagation score, they call it heat. So similar to our approach, they push flow into the network, but then they don't search for clusters, they look for hotspots. So areas in the network with high, we would call it propagation scores. So for the performance evaluation, first we looked how dense are our clusters compared to the other methods. And we had a very significant enriched density for our clusters compared to Prince and Hotnet. But you can argue that because we force our clusters to be connected to at least, that each node is connected to at least half of the other nodes, that this would be cheating. So we also compared it to a relaxed version, so where we don't force the clusters to be connected to at least half of the other nodes, and we're still significantly denser than the Prince method. In the next step, we wanted to check if we have an overlap to known complexes, and therefore we used the database of Quorum and Go, and we compared we compared if we have a significant overlap, and saw that we have, or maybe you can't read it. Okay, I tell you 51.4 for our algorithm, 36.1 for the Prince algorithm, and only 29.9 for HotNet. So as you see, we are able to find those clusters. And in the third step, we wanted to to evaluate our approach further. And as I told you, we only took the manually curated genes per disease. And what we did then is we took for a disease all the genes in the clusters, excluded our priors, and looked so in the newly predicted disease genes, and then looked if they are significantly enriched for the genes that we didn't include in our method. So genes that are associated with the diseases in the 3 databases, but we didn't put in because they are not manually curated. And we found that our algorithm is able to capture a significant enrichment in 34 diseases, where Prince at least had 33 and HotNet 2. So this was the performance evaluation. And now I want to give you 2 examples of such clusters. And the first is for epilepsy. And there are many subtypes of epilepsy, and our prior was containing 97 genes. And in the top cluster contained out of 7 genes and 17 interactions. Okay. And from the prior we had KCNQ2 and 3. These are channels that transmit M current. And M current is important to regulate neuronal excitability in the brain. So if the M current is disturbed, you have a overreaction of neurons, which is one of the things in epilepsy. And what we found, what were predicted are KCNQ5 and CALM123. CALM is the gene name, and CAM is the protein name. So, and we looked then into the literature, what is known about these genes, and found that mice, for example, lacking KCNQ5 channels display an increased excitability of neurons, so similar to what I told you about the KCNQ2 and 3. Also, it was shown that that there is a mutation in KCNQ2 known for epilepsy, which actually expressed a protein which has an alteration in the CAM binding. And that actually CAM overexpression can then restore this channel to a normal function. So what I want to show you is that we're actually able to find proteins of interest with our method. Another example would be intellectual disability. Here's the top cluster containing of 17 genes and 96 interactions. And 11 of these genes are known to form a complex called BUF, which is involved in chromatin remodeling. And 5 of these members are now prior, and 6 we had predicted. This BUF complex is responsible for DNA packaging, which means that what does this DNA packaging means is in the development, the DNA is packed and unpacked, and only if it's unpacked, it's able that genes are expressed. So it's a regulator of transcription. And mutations are associated with mental disorders. Also, we found KLF1, which is annotated as a regulator of erythrocyte development. And mutations are associated with anemia, which is not that interesting for us. But in a recent study, a microdeletion containing also the KLF1 gene was observed in a patient with severe developmental delay, which can indicate that it also has a role in intellectual disability. And with that, I actually end and want to thank you and also Arnon and his boss Rodet. This is me in my office and my boss Erich Wanker. Thank you very much.